Hello everyone, my name is Laura Gonzalez and today I'm going to present new algorithms for accurate and efficient de novo genome assembly for long DNA sequencing reads that is implemented in ngsep software. This work was made at Universidad de los Andes in Bogotá, Colombia. As you may know, DNA sequencing has evolved in the last years and is becoming a common task in different research projects. The CSS protocol of Pacific Biosciences generates long reads around 20 kilovespers with an average error rate below 0.5%, which allows the assembly of complete chromosomes and haplotyping from a huge variety of species. As a consequence of HiFi reads and also ONT reads from Nanopore, new algorithms such as FLAG, Canoe, and HiFi ASM have been developed to achieve nearly perfect assemblies of complex genomes. However, new algorithmic techniques have the potential to further improve the accuracy and computational efficiency to build both haploid and diploid genome assemblies. We present here ngsep 4 as an accurate, efficient, and easy-to-use software to assemble long-read sequencing data. First of all, k-mers among the reads are counted and organized lexicographically. Then, k-mers are ranked based on their counts and a hashing function that is defined as the distance of the ranking to the mode of the counts distribution is applied. Based on that function, we build a minimizers table from the reads to identify overlaps in linear time. A complex undirected graph is then constructed, having two vertices for each read representing the start or the 5' end and the end of the read that is the 3' end. Vertex are saved to collect information useful to identify chimeras and calculate distribution of different features. Overlapping reads are identified by matching k-mers between them following an efficient algorithm similar to that implemented in WTDBG2. After that, statistics collected during this process include an overlap estimation, a new metric that is the coverage of shared k-mers weighted by the repetitiveness of each k-mer, or WCSK, the percentage of the predicted overlap supported by k-mers, and the number of indels. Then, to build the layout path, we implement a machine learning algorithm. In this case, we are using naive Bayesian method, where selecting path edges is assumed as a binary classification problem. So the statistics previously calculated are used as features for our classification. The expected distributions of the different features are inferred from a selected subset of what we call safe edges, which are reciprocal best in both the overlap and the WCSK metrics. And using that algorithm, an edge is then selected if it connects to previously unconnected ends of different paths, avoiding cycles and trees. We run our algorithm on HiFi PacBio sequencing data taken from rice, maize, and the human cell lines CHM13. And then we compare our assemblies with those of Canoe, HiFi ASM, and Fly. And our algorithm ranked first on the rice sample, achieving a median contact length of 30 megabases and a complete assembly of 7 of the 12 chromosomes. In all cases, our algorithm showed better continuity than Fly, but in the more complex genomes of maize and the human cell line were sequenced at lower with depth, they obtain ranked below Canoe and HiFi ASM. However, we obtain genomes that are still highly contiguous and accurate with M50 values above 10 megabases. The statistic numbers of misassemblies also indicate that our assembly has the second best precision for rice and maize datasets, and the Fly showed the fewest misassemblies. Regarding efficiency, our algorithm takes 5 hours for the rice sample and 19 hours for the CHM13 with 32 CPUs. We also run our implementation with Nanopore data taken from Escherichia coli, Saccharomyces cerevisiae, and Drosophila melanogaster, and we compare our assemblies with those from Canoe, Fly, and NICAT. In our case, we ran ngsep using reads corrected with the cat implementation. Our algorithm showed a better performance than Canu on fly in the bacteria and yeast dataset, and a better performance than Canu in the Drosophila dataset. In terms of misassemblies, we ranked second, and fly was the most accurate. So, in conclusion, we present a novel approach to build genome assemblies considering statistical information that can be learned from k-mer abundance tables and distributions of different features. Our algorithm achieved competitive and, in one case, best contiguity and precision compared to current solutions. And finally, we expect to make further improvements in the near future to increase contiguity while keeping precision and efficiency, aiming to produce nearly complete assemblies of mid-complexity genomes and diploid assemblies of human genomes. Thank you.